discussions? If you want, you can use the mic. We have two of them.
Hi, this is for Eda Hanım. What negative effects does inhibiting angiogenesis have on the eye?
That's a really good question, and a lot of people have asked that because so the drugs that are currently available, they're all targeting this one protein, right, VEGF, and that protein is actually required for the normal function of blood vessels as well. So if you take away all of that protein, then all of the blood vessels will go away. So that's why the concentration of these drugs have to be optimized so that, you know, there's still a little bit there, enough to maintain the vasculature, but not take away anything as well. So,
are there any ways to get through to the eye, drug delivery to the eye besides injection?
Yeah, so that's a good question as well, because my technology also would require injection. It's just that it would require less numbers of injections per year. There are other ways, like a lot of people have eye drops, or there are drug-eluting implants, like, you know, polymer implants, whatever, but those have their own consequences. Like if you're using an implant, do you have to take it out? Can it stay in? So yeah, there's a lot of approaches to it, but I'm not trying to reinvent the wheel. I'm trying to make, you know, small incremental changes that can be approved by the FDA. So.
Sure. Alcantarsi, I'm a dentist from Boston, from Forsyth Institute and Harvard University. I'd like to congratulate all the awardees. They're excellent, actually. We were very, very happy to see them over here. My question is back to Eda, because actually, unfortunately, that's the only part that I could understand from the rest of the talks as well. So the question is about the timing of the angiogenesis. How long actually can you shut down the system? Because if you shut it too long, then you will be seeing some side effects for that. What's your comment about that?
Right. So angiogenesis is not supposed to happen in the eye. So in that tissue, in the retina, during development, there's a normal amount of angiogenesis that occurs, and then those vessels are pruned off over time. But in the adult eye, there's not supposed to be any angiogenesis. So whatever new blood vessel growth there is in the retina is not good. So we don't want that. But, you know, in wound healing, you know, when you have a burn, when you have any sort of wound healing, you want it because it promotes, you know, repair of the tissue. But in the retina, we don't want it. So we're trying to knock all that out.
Excellent.
My name is Tayfun Özdemir. I'm from Virtual Aim Incorporated, Ann Arbor, Michigan. And my question is for Ms. Tara Barish. I'm not, you know, medical, but I'm very interested in that. Is, if I can pronounce this, mitochondrial genome defect, if I could define, is that part of the standard fetal screening before birth that we know, some of the DNA screening that takes place?
I'm not actually sure.
Down syndrome and etc.
Right. So usually with that, they just do a karyotype, so they look at all the chromosomes. Since the mitochondria is not part of that, I don't think they look at that. However, if there is mitochondrial deficiencies, then you will usually see it in the mother.
Oh, as part of a disease? Or?
Yeah. So, I mean, there's a slew of diseases that are caused by mitochondrial gene mutations. I can go into more detail. The reason that the mothers are able to kind of survive despite mutations is that they kind of have healthy and unhealthy mitochondria. And so the ratio of those determines how severe the disease is. But you will definitely see symptoms in the mother if she has mitochondrial deficiencies.
Regular, trained physician would know that.
Yeah. Okay. I don't do any of the medical stuff.
Thank you.
Thanks.
My name is Abdullah Çavuşoğlu from the Council of Higher Education, Turkey. I have a question for Erol Akçay. Is it a common thing in U.S. to dedicate a scientific work to the people who are supporting terrorism? Would it be possible for an American or for a Turkish person to dedicate his work to people who are supporting ISIL? Thank you.
Yeah, I'll just say we disagree that they're supporting terrorism. The statement is asking for peace, and it was a peaceful statement, and these people are in jail for just expressing their opinion peacefully.
Are there any technical questions? Please use the mics are there. If you can line up the mics, I'll see you there, and then you can take over.
I'm Umran Inan from Koç University. Alper Ertük, I have a question for Alper. So you discussed a very interesting subject of energy harvesting from vibrations. Of course, you can harvest energy from electromagnetic vibrations in the air from 60 hertz and 50 hertz, and also biological vibrations from the body. Which one is going to win in this competition? Is the vibration that you talked about more prominent?
That's a very interesting question. It really depends on the application and what is available. Like near-field electromagnetic waves and far-field ones. RF is a field by itself, radio frequency wave energy harvesting, but then the power density is not very high. So you can get nanowatts level power output easily, or a few microwatts in most of RF cases. That may not be sufficient for most practical, like wireless sensors that we need. These are typically in milliwatt range for at least my applications. That's why piezo harvesters offer higher power density. And near-field electromagnetic fields, if you have that kind of opportunity to harvest, is going to be stronger to be able to couple with a proper inductor. So it depends on the application. If it's available, I would go for it, definitely.
Yes. The mic.
My question is for Tara Hanım. Does the third mitochondrial donor need to be a woman, and does this need to occur in a surrogate system?
So, yes, it's only done with women. It doesn't have to be in a surrogate system. There are different approaches to it. Either they take out the nucleus from an embryo that's already been fertilized and then put it in, oh, sorry, the other way around. They either take an embryo that's already been fertilized and replace the mitochondria, or take the nucleus from an egg and put it in, take the nucleus out of a second egg and put it in there. But then that can be implanted into the mother. It doesn't matter who's carrying it.
How does the genomic DNA and the mitochondrial DNA communicate? Like, how do they get mixed up together?
Well, communication it's an interesting word for this. Basically, the proteins have to work together. So the proteins that the mitochondria code for have to work with the proteins that the nuclear genome codes for. And like I said, this has kind of evolved to be, I don't like to use the word optimum, but to be optimum. And actually, the mitochondria used to be its own free-living organism. And so it's lost a lot of its ability because it's had to be incorporated into animal cells.
Hello, this is Yasemin Ersin-Holsby. I'd like to congratulate all the winners. Right now I'm adjunct faculty at 2 universities in Chicago. My question is to Tuncay. It's very, very encouraging to see your work. And I was wondering if there are any challenges you can share with us in terms of full commercialization of some of your work.
Sure. We patented the work that I mentioned, taking it to two-dimensional structure. The problem with that is making fully crystalline structures using electrochemistry is a big challenge. So one of the projects that I'm involved in at Harvard University is to use silicon nanowires and then make modifications on those nanowires using electrochemistry. Then, probably since we know a lot of things in terms of silicon research, the use of silicon in solar cells. So these nanowires will be the new generation of them, hopefully. So there's some of the research that I'm involved in at Harvard University in collaboration with Charlie Lieber Group.
Yes.
Hey,
Hi, this is Aybüke, PhD student in digital media. My question to Başak Hanım. You mentioned the, understanding the uncertain behavior. Can you open it a little bit?
So, so what I mean by uncertain behavior is in our conventional communication systems, we have encoders and decoders that operate on standards that are known, and you can predict how they are going to behave up to, given the environment conditions, you can at least estimate. But when it comes to humans, if your encoders and decoders are humans or they're behaving like humans, so they have some uncertain behaviors, they might be influenced by other encoders while they're decoding the messages, for instance. So how do you model this network? How do you communicate under such uncertain behaviors? That's what I was trying to model.
Thank you.
You have a question?
Seda Öğrenci, Mehmet Northwestern University. I have a question for Dr. Asatekin. These membranes in your research sounds quite exciting, and your efforts on commercializing it. And I applaud it also for a female-led a PI going into entrepreneurship and commercialization. That doesn't happen a lot. And I'm curious about whether these types of membranes find uses or will find uses in implantable fashion, implantable into human body.
That will be really interesting. That is something I did not think about before. We have been focusing a lot on separation applications for the simple reason that that is what we know best. And that is where my contacts are with commercialization. So in addition to wastewater treatment, it might be very interesting for the purification of biopharmaceuticals and peptide antibiotics, for example. They could actually be interesting for drug delivery and controlled release. They are not biodegradable polymers, but the same concepts we use to develop these materials, you can adapt them for biodegradable chemistries also. That would be really cool. I would need a collaborator who knows how to do that side of the equation to work on that, but that would be really neat.
Hi, I'm Kerem Joshar from Stockholm School of Economics. I'm an economist, and I would like to ask a question to both Erol Akçay, a scientific question that he deserves, and also Selman, I think, can contribute to this. So you did mention that game theorists and economists work on these issues as well. Do you collaborate with any economist and in potential multidisciplinary work between evolutionary biologists and game theorists and economists? Who's lagging behind? And the same for Selman. Do you collaborate with anyone outside of economics, with network scientists?
So I'll answer first. And yes, I do. In fact, I just had to flash it very briefly, but some of my best co-authors are economists. And political scientists, actually. And so I think it's actually an exciting time to do what I'm doing, because there's actually a great confluence between social sciences, economics, political science, and sociology. Well, maybe sociology is less like science, but it is going on, and biology, and evolutionary biology, and sort of, there's an integration of evolutionary science into social sciences, and what I'm kind of doing is kind of going the reverse way and taking ideas from economics, such as mechanism design, some of you might know that, and applying it to biology. And so yeah, so I do collaborate with economists quite a bit. I even wrote an economics paper and been trying to publish that, so I feel your pain. Yeah.
Well, thanks a lot. I actually don't have collaborators with biology people, but I have finance collaborators and I have network science. Well, there aren't any actual network science departments. Now there are new centers trying to bring all these, different people together. And Rakesh is, for example, founder of the center at UPenn, and Guillermo knows a lot of finance. So this actually requires working together with all these people. But I still did not have the chance to get influenced from the biology side, but I think it's possible. I think there would be a lot of common points how biological networks, neurological networks, and financial networks might actually function. The only little difference, I guess, would be the human factor. And that's at the heart of many economic problems, right? I mean, human behavior is not easy to model, and that's why maybe there is a lot of discord between how humans build systems versus natural systems. So that might be one challenge, but I think there's still a room for that as well.
In the interest of time, I'd like to ask the final question to all the awardees. So I'd like to do one by one if you want to answer. So in your own research, and you're very successful, obviously, what drives you? What is your source of motivation, inspiration? And what's next for you? What do you want to achieve in the next 5 years? If you come up and use Mike to answer those two questions to wrap up the session, that would be great. Who wants to start? We'll start with the most senior one then.
Please.
Maybe the senior faculty this time.
The order in which we have the order.
So, yeah, wow, big question. So I'm sort of fundamentally interested in sort of how biology and biological and social systems are organized, and how sort of they're organized without a designer and without sort of just the parts sort of doing whatever is best for them. And so that's sort of my overall motivating question, and I kind of find myself drawn to questions that have to do with, how do you achieve cooperation when there's conflict of interest, and how do you sort of set up, how does natural selection set up these systems, and how can we learn from it maybe to better the human condition. And yeah, so in the next 5 years, my overall goal is to sort of better integrate that four sort of quadrants of social evolution theory, especially the processes that sort of structure social networks and social interactions with how individuals, with sort of the decision-making processes that go on in individuals' brains. So there's developed new theories of that can hopefully be tested in the field, and and maybe even lead to sort of applications in for human society.
I think the short answer is curiosity. So my research is very curiosity-driven. I did my master's back in Turkey, got my degree in 2006, worked with Dr. Nevzat Özgüven. Currently, I think he's the vice president. That was entirely on vibration, avoiding vibrations, and I came to US right now. We're exploiting vibrations. So learning new things and curiosity is the main drive. And what is next? I've been fortunate to just get tenured at Georgia Tech, and at a place like that, you have many opportunities to learn new things from different collaborators. So I would like to just go into more high-risk areas.
What drives me is sort of two things. The first is making a difference for a world that is more sustainable, so protecting our, as I said, water resources, but also energy, and they are very interlinked. And being somebody who is especially interested and who knows most about technology and science, that is my toolkit to make a difference there. So that is what I work on. And honestly, I get really excited about cool science. So you have to have a little bit of both, I think. In terms of what is next, we have been working a lot on water treatment membranes. I think I am really excited to see how our chemical separation, chemical processing membranes turn out. It is less sexy, but I think it is a bigger step in terms of bringing a new technology. And also, I think it is really exciting to see all my students grow and graduate. So that is the other thing that is next for me.
I think for me, I'm really into learning new things, and there's a lot of opportunities when we are in science. We can learn everything in the lab. And not just learning, but also teaching someone is also very exciting for me. So this semester, I'm lucky to teach at Harvard University to undergrads. And during my teaching experience, I learned a lot from them. So it goes both ways. So I think for the next 5 years, I'm planning to have my own lab and keep learning about research, and also teaching and having a good generation for the future.
Well, I guess looking into the future, I would like to establish myself as an independent researcher and develop my own research group and do science eventually. And what drives me most in my research is the
is the fundamental understanding of how things work, basically, and how, from that understanding, how we can use, like, improve things for the greater good of the people and science. So, and the most importantly, I would like to be, help educate in my independent career, help educate the next generation of scientists.
I think my biggest motivator was improving healthcare for patients. I hope that with my presentation, you guys saw that it is a translatable drug, like it can work. And some of the biggest, like the coolest memories from grad school were actually interacting with patients who told me that they really needed this drug. So that was really the biggest motivation. And next step, I'm just starting my career at Exponent, the consulting company, so I'm excited about that as well.
For me, my biggest motivating factor is fascination. When I first learned about evolution and how it's shaped pretty much every living organism around us, I couldn't stop thinking about it. And so that's what I've pursued in my studies. And even though I showed my work in a medical context, evolution and ecology are really what my passions are. In terms of the future, I hope to continue my research and keep finding out cool things.
This might be a little bit of an unorthodox answer, but I think academic lifestyle is just very cool, very comfortable, and it's a great life. And for the future, well, I mean, I'm in a special situation doing economics and finance, so I actually am interested in, after learning sufficiently more, getting incorporated into maybe policy work, because at the end of it all, I think we have to give back to society, and this might be a direct way of doing it. And I'm really eager to be able to actually set up links between Turkish academic world and U.S. academic world. I think, well, I'm not saying we should reverse the brain drain, but there should be more links between Turkey and U.S., and I would like to contribute to that.
My main motivation was also curiosity, which was already being told. So I was really lucky to be given the opportunity to work on the problems that I like. I have always been interested in how things work. When I was doing my undergrad studies in Middle East Technical University, I was in electrical engineering department, and I was also interested in social networks. I was reading books about sociology, but then everyone told me, oh, electrical engineering and sociology, how these two things can go, can combine. And now I am working on problems modeling social networks from an information theory perspective. So I think curiosity and finding the problem that you like to work on is the main drive for me. And another one other thing that is really interesting is to see how abstract ideas like information, so vague, how they can become quantified. How can we really quantify information? How can we use it in real systems? I think this is really nice to bring abstract ideas like this in practice. Thank you.